everybody, and thank you for joining us for night 2 of our Charleston Fellow Bachelor Essay presentations. Tonight we've got 4 wonderful students lined up for you to tell you all about the research they've been doing this last year or even more here at the College of Charleston. So our first speaker tonight is going to be Brogan Brown. Brogan is a biology major, and she's been working with Dr. Camilla Rochelle at the Medical University of South Carolina. And her project is entitled A Sex-Differentiated Study of Rodent Empathic Behavior, Ultrasonic Vocalizations, and Oxytocin Activity. So let me cue up your presentation, Brogan.
All right. So what is empathy? So this was kind of a guiding question when we first went into our research. We've defined empathy as a pro-social higher order emotional process in which an individual is able to understand another's internal state. But you're probably thinking empathy is a little bit difficult or maybe abstract. How do you study that on a more scientific scale, a more scientific way? And the way that my lab and I went about this is through something called the perception action model. The perception action model is about the most evolutionary, basic, conserved way to think about empathy. And it's described in this image right here. And this will kind of be the basis of the entire project and the activity that we put our rodents through. So first we have our target, and the target experienced some distress. This could be experiencing pain in some way, discomfort in some way. That's the easiest first thought to kind of think of. The target experiences distress, and there's an emotional transfer to an observer. And the observer shares in that effect. The observer can see that the target is experiencing it, and the observer then can aid to reduce the distress and ultimately feel relief for itself. So at the end of the perception action model, we have a target experiencing distress, the observer aiding, and then ultimately both individuals feel the relief of being out of that situation. And empathy is really important because we know that empathy impairments are present in some neuropsychiatric diseases such as autism spectrum disorder and substance use disorder. All right, so the way that we went about studying empathy was through a novel 3-chamber task. And as you can see, I have that image of the PAM model there on the left. And I'm going to walk you through what we had our rodents do to align with the PAM model. So here we have our target animal. And this animal, this little rodent, he is submerged in a pool of water. And if you know anything about rodents, they do not like that. So he's experiencing some distress, squeaking, maybe splashing around a little bit. And then he has the observer here on the left that is able to witness this animal's distress through the plexiglass. And the observer animal is ultimately able to pull a chain which releases the wet animal into a safe and dry compartment. So as you can see here, we have created a model that aligns with what I just described with the PAM action model. So the next thing that we were interested in is I've said a few times like, oh, the animal is experiencing distress, the animal is feeling this. Well, what's the scientific way to kind of peer into the animal's mind and know what they're thinking, so to say? The way that we do this is through ultrasonic vocalizations. These are essentially rat squeaks that are too high frequency for our ears to hear, and they can give us an indication for how the rodents are feeling during this task. So what we did in our study was we attached a microphone to the 3-chamber task, so we can listen in on the squeaks and the communication between the 2 rodents. We know from previous research that squeaks of lower frequency are distress calls, and squeaks of higher frequency are social calls. So this is something that we use to guide kind of how we think about the animals during this task. The next thing that we're interested in is looking into the brain and what's happening in the underlying processes in the brain. And we mainly looked at oxytocin. And oxytocin is a neuropeptide, and it's primarily known for its role in maternal behavior, such as uterine contractions and milk letdown. But what we're really interested in, and what has really been studied recently, is the way that oxytocin can act to promote social behavior, such as bonding and social interaction. We also know there is some evidence that suggests that oxytocin may be regulating the underlying processes of empathic behavior, such as emotional recognition and accuracy. And we know this because oxytocin is synthesized in the paraventricular nucleus of the hypothalamus, or the PVN. And the PVN projects to several brain regions that are involved in social-emotional processing. So basically, that's what this image is describing here. We have the PVN with the making of oxytocin, and it's extending to all these other regions that we know are very important for understanding social situations and emotion. So what we did, this is a little model of a rat brain, and where the PVN is in the rat brain. In our study, we tagged the oxytocin that we see activated during the empathic behavior task in these rodents. So our 3 research questions: does sex impact empathic behavior? Does sex impact ultrasonic vocalizations during empathic behavior? And then, does sex impact oxytocin activity in the PVN during empathic behavior? So for our methodology, we ran a cohort of male and female rats through the 3-chamber task, and we looked at 3 things. The first thing that we looked at was latency. This is essentially how long in seconds did it take for the rodent to learn to pull that chain and release his friend. So we timed that. The next thing, we looked at ultrasonic vocalizations using a program called Deep Squeak, which is a deep learning program that can identify some of the vocalizations. And then finally, we performed immunohistochemistry and analyzed oxytocin activity. So first thing we found, as you can see here, this describes the behavior among the sexes, males and females. So we have the latency, how long it took for the observer rat to pull that chain, and then we have days. So over the 10-day period, both the males and females learned the task. There's a significant difference, and they learned essentially to pull that chain faster and act empathically. But we also see here is a sex difference between the males and the females, with the males performing actually better, you can see in this white dot line. But one thing that we found really interesting was this region of the data called early acquisition. And right here, we see no sex difference between males and females. This time period is when the animals are kind of learning what the task is, how to do it. And we looked a little bit deeper into this region because we didn't see that difference that we saw in the rest of the graph. As you can see here, this is the latency for those first 3 days, pretty much exactly the same. So we looked into that early acquisition time period, and we found this really interesting binodal graph of the call frequencies that clustered around 2 ranges. It clustered around that distressed range, the 30 to 40 kilohertz, and then it clustered around the social range. And we pretty much saw that even though we see maybe some variation in those peaks, we see that with each animal regardless of sex or their role. But we did see one kind of standout data here with the female targets. This would be the female animal that was wet and the wet part. The female was calling out a lot more distress calls than the male target or any other role, and a lot less pro-social calls. The next thing we looked at was the oxytocin activity in the paraventricular nucleus, or the PVN. And this is actually images that I took. We have the oxytocin in the paraventricular nucleus. So we tagged for FOS, which is right here, which is just a general activity marker. It lets us know what neurons are active. And then we tagged for oxytocin. We overlaid those images and counted the overlap. So which of the active neurons are oxytocin? And as you can see on this graph here, we have the percent overlap. So when those green and those red overlap. And we have the home cage control. And this is a condition where the rodents just sat in their cages, they didn't do anything, maybe just like ate and slept and ran around just like normal rodents. And then we have the empathy animals that we ran through the empathic behavior task. And we see for the males, we see a higher percent overlap after the empathic task, but we see the opposite for the females. We see actually a decline in that overlap of oxytocin. So our results. So we found with the male and female rodents, there is a sex difference across all trials of the empathic behavior task favoring the males, but there was no sex difference in early acquisition in those early phases, those early trials. For the ultrasonic vocalizations, we found the bimodal frequency of calls, and then we found an increase in distress and a decrease in prosocial calls for target females in early acquisition. And for the oxytocin, we found a possible convergent sex effect in early acquisition. So the males and the females were performing the same, but we saw an increase in oxytocin activity for the males and a decrease in oxytocin overlap in the females. So our next steps moving forward is we will perform an experiment where we inject both male and female with oxytocin before entering into the behavior task and see if that oxytocin injection modulates the empathic behavior during early acquisition. And I would like to thank the Rochelle Lab and my on-campus faculty member that oversaw this project, Dr. Korey.
Any questions from the audience?
Not at the moment, and we're having some camera issues with the focus, but we're
Okay.
trying to get those sorted out.
Can I ask a question while you get?
Absolutely.
Okay, awesome. So, nice job, Brogan. So I was trying to see, I couldn't see all the data because I was trying because of my angle. But I had a question about the empathy and what your thoughts are on whether or not that's something that the animals learn or that they're born with. So, or at what stage do they develop that? And so I understand that they had to learn the task, but that has nothing to do with whether or not they were empathetic before that, right? So kind of what age were you looking at? Have you thought about if it's something that they have to learn or not?
Okay. I think just from, there's been a lot of studies that look at rodent empathy. I do think that it's something that the animal possesses, at least on the most basic level, the perception action model that I was talking about. At the most basic level, they possess empathy. We've tested our animals in adulthood, so I don't know the exact amount of days or weeks that they were.
But they were adults.
But they were adults. Yes.
Has anyone looked at earlier, I don't know how you could even do that. And then, I mean, obviously you have to have a different behavioral paradigm to use.
Yeah, not that I've
neonatals.
seen
Okay.
with the prenatal or the natal younger rodents. Our study, there's a decent number of studies out there that look at rodent empathy. What makes this model interesting is the 3-chamber part, because a lot of them will just do 2 chambers, and then they have the confounding variable of social interaction. So I definitely think that empathic behavior is something a rodent naturally has. It was just a lot of these models have these confounding variables, so this model really was novel in that it took that out of it.
No, I mean, that'd be something certainly interesting to kind of look at. The other question I had to do with the male and female differences, which I think is interesting how they're differentially activating that oxytocin system. So I'm assuming they've all been gonadectomized, like male testes removed, female ovaries removed?
Yeah.
Okay. And so would you think that if that wasn't the case, that you would see like differences, in the females across their estrous cycle, or, anything in that regard? Has anyone looked at pregnant dams?
Oh, that would be interesting.
Yeah.
I think there would be a difference just because they know that sex hormones interact with oxytocin. It would be interesting to look at, especially in pregnant dams.
But right now these have no sex hormones.
No sex hormones, yeah.
That's
If you
really,
go to the data at the final slide I have on there, click 2 more, or go forward.
sorry. Oh,
To
gotcha.
the next one.
Oh, okay.
So this acquisition curve for the males and females, do you see day 5?
Yep.
The males and females are actually very strikingly similar the thing that throws them off is day 5. We've done lots of these trials just to get our N up, and the behavior is almost exactly the same. It's just there's like high performing females and high performing males, and there's low performing females and low performing males, and those low performing females perform really poorly. So that's what has driven that day 5 up. But I honestly think that as we do more trials, we might get a curve that has no significance between the two sexes.
But you still have the differences in the oxytocin neuron activation. So what would be interesting is to correlate that number, that overlap, those co-labeled cells with the individual animal behavior. So are your low behaving animals, your low empathy females, how do they fall on this range with regards to the oxytocin level? So, all right. Cool. Any other questions?